Okay, so good afternoon, everyone, and welcome back after our week away from human physiology. I hope that you had a good time away, that nothing too sad or depressing took place while you were away. I can't think of anything sad or depressing that would have happened, so hopefully that's good. Just a couple of housekeeping things here before we dive into our lecture. So hopefully you have not forgotten that we are currently in the midst of term quiz number 4. So that opened up yesterday at midnight, and it closes this evening at 11:59. So make sure you take some time to log in and complete that term quiz before the end of the day. I will then, as I always do, release those grades tomorrow morning, and that'll kickstart our 3-day rebuttal period for anyone who's interested in that process. Other than that, we have our bonus deadline for the short communications coming up. It's 10 days from now, I guess, on the 14th of November. There are some groups that are getting very close to the end of that short communication 1, so that's a good sign. There are other groups that have not yet started the project, so we're kind of everywhere in between. Certainly if you're a group that hasn't started and maybe you just need some assistance from my end to help kind of get you going, let me know. And I'll just again remind that if you've had a submission sitting around for more than 3 business days, don't hesitate to email me, and I'll try to take care of it and get it evaluated so we keep things moving along. Any questions about things before we continue on with our lecture? Everybody's good. Okay. So before we left for the break, we were learning about synapses, and in particular, we were talking about electrical synapses. And one of the things we learned about electrical synapses is they're very often found in places in the nervous system where, well, one thing we talked about was speed. That was actually a previous slide, that we have no synaptic delay across an electrical synapse. So where we need speed, we'll often find electrical synapses. We also find electrical synapses in places where we need synchronicity among neuronal populations. We had the example here of these gonadotropin-releasing hormone neurons. We said that when gonadotropin-releasing hormone is released in these discrete pulses, as opposed to a continuous release, you get better responsiveness from the pituitary in terms of releasing luteinizing hormone. And so to get these discrete pulses, you need all of those GnRH neurons to be kind of excited all at the same time. So we noted that basically, we noted that there's a good expression of connexin genes in the neurons that make gonadotropin-releasing hormone, and that means that these neurons are all connected so that when one gets excited to stimulate its gonadotropin-releasing hormone, the electrical synapses allow for the simultaneous excitation of all the other neurons in that population so that they ultimately will all release their hormone at the same time, and that's how we get these discrete pulses of hormone release as opposed to, like I said, as opposed to having more of a continuous hormone release. The third and final thing I want to talk about with respect to electrical synapses, which moves us on to some new content here today, is that we find electrical synapses are very common in the embryonic nervous system. They're actually perhaps the predominant synapse within the embryonic nervous system, and then as the nervous system continues to develop, they will give way to the chemical synapses more so. Why do we have electrical synapses so predominant within the developing nervous system? We think it's because of the ability that electrical synapses have to allow for molecules, again, to easily move through populations of neurons. And as we're going to talk about here in a second, we have these, what I'm calling here, guiding cue molecules that are important for helping nervous systems figure out, what should I develop into? Different parts of the nervous system have to develop into different nervous structures. How does different parts of the embryonic nervous system know what it should develop into? Should I become a brain? Should I become a spinal cord? Should I become a sympathetic ganglion? Should I become this or that? How do they know? Well, it's actually all facilitated by these guiding cues. And so just to kind of give a little bit of background here first, so here I have a picture of a developing human embryo, and in yellow you can see the neural tube, right? So the neural tube is what gives rise to the brain and the spinal cord. So you can see here that at the head end, the neural tube is elaborating into the various brain regions, forebrain, midbrain, hindbrain, and then in the more posterior regions, the neural tube is developing into the spinal cord. But again, we have to think to ourselves, okay, here's this neural tube that forms. How do the different parts of the neural tube know where they are in this embryo and therefore what they should become? Like, why don't we end up with brains in our asses here? If things go wrong, we could. So these guiding cues that I'm going to introduce you to in a moment, they are responsible for communicating to the neural tube where the different parts of the neural tube are located in the embryo to ensure that we actually develop the brain in the right region, brain structures in the right part of the brain, spinal cord in the right spot, et cetera, et cetera. Now, just to talk a little bit about the formation of this neural tube, you're probably at least a little bit familiar with kind of the three embryonic germ layers, the ectoderm, the mesoderm, the endoderm. We're looking at a little bit of that here. So here you can see a part of a developing embryo, and you have the ectoderm here on top, and a few bits of mesoderm underneath, and they're not showing us any endoderm because it's totally irrelevant to the conversation at this point. But what you will notice here is that at some point in development, part of the ectoderm differentiates into what's called the neural plate, or the neural ectoderm. And what will happen to that neural ectoderm or neural plate is it will invaginate, right? So it'll kind of move inward within the embryo, and then eventually it will disconnect itself from the overlying ectoderm, and this is how you end up with the neural tube. Okay, so the neural tube is derived ultimately from the ectoderm, specifically this neural ectodermal region that crops up during development. And so what I really want to highlight here is how the neural tube, A, how it forms, but B, just to note that the neural tube then is sitting between this ectoderm here, and then underneath of the neural tube is the notochord, which is basically kind of the skeletal, structural element of the embryo, if you will. You'll recognize that term notochord if you've ever either studied human development, I suppose, or if you've studied lesser vertebrates, Amphioxus or something, where they actually retain the notochord as their kind of only skeletal structure. But I digress in any case. So just want to give you a sense of where this neural tube is located within the embryo. And it's important to note its location because these guiding cues that I'm talking about here in a moment, they're actually initially produced by the ectoderm that's kind of dorsal here to the neural tube, and they're also produced by the notochord, which is ventral to the neural tube. These two structures are initially responsible for synthesizing these guiding cues, and then eventually over time, the neural tube will just start producing its own guiding cues to facilitate its development. Now, what are these cues? Now, the first thing you're going to notice here is I swapped out the figure that I originally had in the notes, mostly because I had lots of time over well, not lots of time, but as I was going over things during reading week, I was like, something's not right with this figure that I have here. It had it had the wint flipped around, so I replaced it with a figure that's actually a more accurate figure here. But in any case, these guiding cues that I keep hounding about here, the three of them are mentioned here. So one is BMP, which stands for bone morphogenic protein. The next one is SHH, which actually stands for Sonic the Hedgehog. So that actually got its name because the postdoc who discovered it, apparently his daughter was a huge fan of Sonic the Hedgehog, so they just gave it the name Sonic the Hedgehog as kind of an homage to this legendary video game character of the 90s. And then the third player here, the third guiding cue, is WINT, which stands for the wingless integrator. And it got its name in kind of a weird way. It's because two different lab groups had kind of separately discovered the same protein and given it different names. And once people realize, like, oh, this is actually the same protein, they just kind of combine the names together. So the wingless integrator is what WINT stands for. In any case, what we can appreciate, what this figure is trying to highlight for us here, is that the BMP, so this bone morphogenic protein, this is initially produced by, if I go back, this is initially produced by the ectoderm. So it's being produced on the dorsal side of the neural tube. And then later, the neural tube will actually start producing it itself from the region called the dorsal roof plate here. So BMP is a guiding cue that's being produced on the dorsal side of this neural tube, either by the overlying ectoderm or eventually by the dorsal roof plate of the neural tube itself. And then because you have all these electrical synapses that connect all the neurons of this neural tube together in this embryonic state, this BMP that's produced on the dorsal side of the neural tube is able to diffuse through all the neurons within that neural tube, and you end up with this gradient that forms, right, where the BMP concentration is very high on the dorsal side of the tube and gets gradually lesser and lesser as it diffuses from neuron to neuron through the neural tube. And so this concentration gradient for BMP, which is strong in the dorsal region and weaker in the ventral region of the neural tube, if you're a neuron that is within this neural tube, you can kind of use the concentration of BMP to gauge where you're located on a dorsal ventral axis within this neural tube, right? Because if you're getting a strong BMP signal, then you're probably somewhere near the dorsal surface of this tube. And if you're getting a weak BMP signal, then you're probably somewhere closer to the ventral region. And so depending on the strength of that BMP signal, you can gauge where you are on this dorsoventral axis. And then from that information, you can determine, oh, this is the kind of thing, structure that I'm supposed to develop into because I'm in this location within the neural tube. Similarly, we have that Sonic the Hedgehog here. So this is initially produced by the notochord, which if I go back again, is ventral to the neural tube. And then over time, the ventral floor plate, so the kind of bottom ventral surface of the neural tube will take over and produce that Sonic the Hedgehog protein. And then again, that Sonic the Hedgehog protein will diffuse through all the electrical synapses, through all the neurons within the neural tube. And now you'll have this, again, sort of dorsal ventral gradient for this Sonic the Hedgehog protein. And so the closer you are to the ventral surface of the neural tube, the stronger that Sonic the Hedgehog signal is going to be. And as you get closer and closer to the dorsal surface, the weaker that signal is going to be. So we effectively have these two dorsoventral signals here, BMP and Sonic the Hedgehog. And again, any neuron within this neural tube can assess what's the concentration or strength of the BMP signal I'm receiving, what's the concentration or strength of the Sonic the Hedgehog signal I'm receiving, and they will be able to know where they are physically located within that dorsal ventral axis. If that's not being clear to you, think of it this way. Let's imagine that I just kind of grabbed one of you from the room here and threw you into this big octagonal. Are we an octagon? Yeah, right? Yeah, octagonal surface here. So threw you out here somewhere, right? I'm going to stand at one end and be talking, and I'll have him at the other end talking. And you've got your eyes closed, and you don't know where we threw you into this room, but you do know that he's on one end, and you know that I'm on the other end. If we're talking, you could say, oh, well, who can I hear more clearly? That's going to give me a sense of which side of the room am I on, right? If I'm right in the middle, I'll probably hear them equally. I'll know I'm in the middle. If I can hear this guy over here talking louder, then I know I'm probably closer to here. And if I hear him talking louder, I'm probably over here. So I'm able to assess my position without being able to see anything just based on the strength of those two voices. Just like here, I can use the strength of the BMP and SHH signal here to gauge where must I be in that dorsal ventral axis of the neural tube. The third signal here is WINT, and so this is produced predominantly by the posterior end of the neural tube. And so it's going to be produced in the posterior end and gradually diffused through all the various electrical synapses to the anterior end. And so again, the strength of the Wnt signal will be much greater in the posterior end and much weaker in the anterior end. And therefore, I can use that gradient to figure out, well, where am I as a neuron within the anterior-posterior axis of this neural tube, right? If I'm getting a very strong Wnt signal, then I know, okay, I'm somewhere back here. If I'm simultaneously getting a strong BMP signal, then I know I'm up here. If I'm getting a strong Wnt and a strong Sonic the Hedgehog signal, I know I'm here. You know, if I'm getting a weak Wnt signal and a strong Sonic the Hedgehog signal, I know I'm here. If I'm getting a weak Wnt signal and a strong BMP signal, I know I'm over here, right? So I can figure out where I am in this kind of whole neural tube, both kind of dorsal-ventral and anterior-posterior, by being able to assess the strength of these signals. And so where the electrical synapses, again, come in here is they simply, again, provide that cytoplasmic connection between neighboring neurons. And so it allows for these Wnt and BMP and Sonic Hedgehog signals to very easily kind of move throughout the entire neural tube in order to establish these signaling gradients. Okay, so that's enough said about electrical synapses. I want to move on now to chemical synapses, which again tend to predominate, especially within the adult nervous system. And so I just begin here with a bit of an overview of how these synapses work, and then we'll kind of go into more of the details thereafter. We already know the basics of chemical synapses. We know that you have your presynaptic neuron here, and then it has these neurotransmitter-filled synaptic vesicles. We know that they're eventually going to undergo exocytosis to release their contents. And then on the postsynaptic membrane, you've got a receptor for that neurotransmitter, and when that neurotransmitter binds to that receptor, that should lead to some kind of graded potential occurring in that postsynaptic cell. So, in terms of the details here, how do we actually trigger the release of the neurotransmitters? How do we trigger the exocytosis of those synaptic vesicles? The answer is that an action potential, it's over here, an action potential is going to arrive at this axon terminal, and we know that the first phase of every action potential is a depolarization phase. So we're going to have this depolarization arriving at the axon terminal, and present in the axon terminal, present in this postsynaptic neuron's axon terminal, are these voltage-gated calcium channels. Right, we haven't talked about these yet. We've talked about voltage-gated sodium and voltage-gated potassium channels, both important for action potentials. But here we have voltage-gated calcium channels, and so they are tripped open by depolarization. So when we have the action potential arriving here in the axon terminal of this presynaptic neuron, the depolarization phase of the action potential triggers the opening of these voltage-gated calcium channels, and then that allows for calcium to come into the axon terminal. What the calcium will then do is it will bind to a protein that's embedded within the synaptic vesicle, and we'll go into these details in a second, and then the binding of that calcium to that protein is what triggers the exocytosis process to occur. And so you can see that here, where the calcium comes in, basically interacts with the vesicle, leading to exocytosis. And again, they're showing some of that neurotransmitter that's been released, binding to a receptor on the postsynaptic membrane, and then that will ultimately trigger the opening, or closing perhaps, of some gated ion channel, leading then to a change in the membrane potential in that postsynaptic cell, which we call a graded potential. They also highlight for us here, and we'll go into these details later in the week, that at some point you do have to clear the neurotransmitter out of the synaptic cleft, lest you have continuous stimulation on this postsynaptic neuron, forever and ever and ever more, just because you released it once. And so they do kind of highlight here the three ways that we can clear that neurotransmitter out. One is the neurotransmitter can just be taken back up by the neuron that released it. So there are reuptake transporters. These are not only found in the neurons, but they're also found in some of the glial cells, especially the astrocytes. right? Astrocytes can take up neurotransmitters from the cleft, help kind of clear out those neurotransmitters. There are often enzymes present either on the post or sometimes the presynaptic membrane that can actually enzymatically degrade the neurotransmitter and get rid of it that way. Or lastly, that neurotransmitter can just diffuse away from the synaptic cleft. We're going to go into more details about those mechanisms later in the lecture, but just because they're here, I thought I'd briefly mention them. So those are the basics of how a chemical synapse works. Let's now dive into some of the details here. Starting with the voltage-gated calcium channels, because we said that when you open these channels, calcium is going to come into the axon terminal. But I feel like we should think about, well, why does that happen? Why doesn't calcium leave the terminal instead? What's driving it in? So this is taking us back to stuff we talked about much earlier in the course when we were talking about electrophysiology. We didn't focus on calcium at that time, but we're going to focus on it now. And so what you'll notice here is, in terms of calcium concentration, there's virtually no calcium in the intracellular fluids, right? And actually, again, at the very end of the electrophysiology lecture, we kind of talked about why that's the case. Because when you have calcium present at a certain concentration within the intracellular fluids, we saw that it actually triggers cell death, right? We talked about it in the context of multiple sclerosis and how you have this influx of calcium that eventually takes place that basically kickstarts a cascade of events that leads ultimately to the neuron's death. So we have to keep the concentration of calcium in the intracellular fluid very low to protect the cell from apoptosis. And so how is that accomplished? Well, I think we're a little bit familiar with this now. One is that there's a calcium ATPase that is directly using the energy of ATP to power the pumping of calcium out of the cell. And we became familiar with that again back in the electrophysiology lecture. So this is not new information. And the other player is the calcium-sodium exchanger that we got introduced to last week. The way that it works normally, right, not in the multiple sclerosis case where it goes backwards, the way that it works normally is we are pumping sodium out of our cells using the sodium-potassium ATPase, and then as sodium leaks back into the cell through this exchanger, we couple that influx of sodium to the efflux of calcium. We basically use the energy stored in that sodium gradient, the energy stored in the resting membrane potential, if you will. We use that energy to drive the outflux or the efflux of calcium. So these are the two mechanisms working together to keep the intracellular calcium level extraordinarily low in our cells. And therefore, the concentration of calcium is much more abundant in the extracellular fluids. Now, if we come back to our Nernst equation, which we're familiar with too, we know that we can work out calcium's equilibrium potential by using the Nernst equation. And so that's what I've done here, right? I've put in the exact same constants, my gas constant and Faraday's constant, same 37-degree temperature we used before. Calcium's got a +2 charge on it, so that's why the 2 is there. And then I've just put in my extracellular and intracellular concentrations of calcium, just kind of eyeballing them off the graph here. And what you'll notice is when you do the math on this, you end up finding that the equilibrium potential for calcium is a very positive value, right? I've got it worked out here to +52. So this makes sense now in terms of, well, why does calcium come into the axon terminal when there is this depolarization? Even at the peak of an action potential, we typically get only to around, say, +30, +40 for our peak membrane potential. That's still going to be more negative than calcium's equilibrium potential. It wants the inside of that cell to be +52 millivolts relative to the outside. So even when we're at something like +30, +40, +45, who cares? We're not at the equilibrium potential that calcium wants. And so because it wants the membrane potential to be more positive, that positively charged calcium has to come into the cell through that open voltage-gated channel in order to get to equilibrium. So that's what's driving the calcium influx here when those voltage-gated calcium channels are opened. What we have to think about next now is how exactly does this influx of calcium into the axon terminal of this presynaptic neuron, how exactly does that cause the exocytosis of these synaptic vesicles to occur? Well, before we get into that, we have to first acknowledge that there are actually 3 different pools of these synaptic vesicles present within the axon terminal. So yeah, we have something called the 3-pool model. So the first pool I'll direct your attention to is this one down here that's called the readily releasable pool. These, number one, this is the smallest pool, as you can see from this figure. Only about 5% of the vesicles are part of this pool. And the reason we call it a readily releasable pool is because these vesicles, as you can see here, are docked already to the presynaptic membrane. They are docked on to that membrane, and so they are just waiting for a calcium signal, and then they can just move that very short distance from where they're docked to the presynaptic membrane, merge with it, and then release their neurotransmitter content into the synaptic cleft. So that's why they're called readily releasable. Actually, came across a cool analogy that I'm going to kind of work in here, which is to think about these pools kind of in the context of a vending machine, right? Go to the vending machine to get a chocolate bar. The readily releasable pool would be akin to the chocolate bar that's right in the front, right? The one that you know you're going to get if you push that button, right? It's right there ready to be released. It's just waiting for you to put in your coins or tap your card, push the button, and it's coming to you, and you get chocolatey goodness or some other kind of goodness, whatever kind of goodness you like, right? So that's our readily releasable pool. It's docked, ready to go, ready to be released, just waiting for a calcium signal. Your second pool is a little bit further back, called the recycling vesicle pool. And what happens here is as soon as these readily releasable vesicles undergo exocytosis, vesicles from the recycling pool can rapidly come in and dock in their place, right? And so when they talk over here about fast, right, this fast relationship between these two pools, that's what they mean. That as soon as that readily released vesicle is released, as soon as it undergoes exocytosis, there's a recycling vesicle pool that can rapidly come in, dock to the membrane, and take its place. Going back to the vending machine analogy, you put in your coin, you push the button, a chocolate came out. What happens? Another chocolate moves forward. It's ready to be released if you want to buy another bar. So that chocolate, all the chocolate that's kind of in behind the front chocolate, that's akin to these recycling vesicle pool vesicles, right? They are right there, ready to come in and replace the one that was released, and they can kind of do that rapidly, okay? The third pool here is called the reserve vesicle pool. It's by far the largest, as you can see. About 85% of all the vesicles belong to this pool. And what happens here is when you have started to use up all of your readily releasable and recycling vesicle pools, the reserve pool can kind of then release vesicles that can replace those other two pools, but it does so only kind of slowly. Right? And so the idea here is that, especially in cases where we have like this neuron being stimulated kind of, at high frequency, very intensely stimulated, and we got to release a lot of neurotransmitter to communicate that intense stimulation onto the next neuron, we need this huge pool of neurotransmitter vesicles that we can then, recruit to release their neurotransmitter, right, to have lots of neurotransmitter be released because of that intense signaling that this neuron is experiencing. But we also don't want all that neurotransmitter to just be released all at once. If all those reserve vesicles go rapidly to the membrane and release their contents, you're going to pretty quickly run out of neurotransmitters. So the slowness of how the reserve pool vesicles make their way to the membrane, that slowness is intentional. That slowness is a way to say, okay, yeah, this neuron's being stimulated pretty intensely. We got to release a lot of neurotransmitter, but let's release it at a slow, steady pace so that if this is a prolonged nervous stimulation that we're experiencing here, we're actually going to have enough neurotransmitter to kind of release during that whole time, right? You don't want to, blow the whole load of neurotransmitter here in a millisecond. So we have lots of neurotransmitter to release, but we want to release it slowly over time if there is this prolonged intense stimulation. Going back to the vending machine analogy, it's kind of like the reserve vesicle pools are like the candy that's in the stockroom. So when the vending machine gets emptied out, you've exhausted all of the candy that was in the vending machine, you've used up all the readily releasable and recycling pool vesicles, someone has to go and restock the vending machine, right? And that takes time, right? There's not a guy there with a box waiting for you, he waits till the machine is empty or more or less empty and then goes and gets the box in the stockroom and brings it out and fills the machine back up. So it's kind of a slow process at this point of releasing more chocolate bars, right? But that's to ensure, this is where the analogy kind of falls apart, because pretty clearly if you're selling chocolate bars, you don't give a shit how fast those chocolate bars go sold. But at some point it's like, we don't want to run out of chocolate bars. We want to make sure everybody who comes along can get one. We don't want them to be like so rapidly refilled into the machine that they just get emptied again. Yeah, the analogy falls apart at that point. But you get the point, I hope. That this is the reserve pool, right? This is the pool of neurotransmitters that we're going to release if the neuron is experiencing intense stimulation for a long time, and having it release only slowly is a way to ensure that we don't, again, blow the whole load of neurotransmitters in a short period of time, that we can actually have prolonged neurotransmitter release over this extended period of time that this neuron might get stimulated. Of course, I will point out here, at some point, you could just run out of neurotransmitter. That happens, right? I remember when I was doing a postdoc. When I was doing my postdoc, I was teaching a course at Western, and we actually had a tutorial of some kind. I can't remember the details, but it was a tutorial where it basically showed data about muscle fatigue. And it showed that the fatigue of a muscle is sometimes not caused by the muscle itself, getting tired and unable to contract. In some cases, muscle fatigue occurs because the somatic motor neurons that are stimulating that muscle run out of acetylcholine to stimulate it with. And they actually would show in the tutorial in the data that we had that once the muscle had become fatigued, if you directly stimulated the muscle electrically, it would contract no problem. But the problem was the acetylcholine had run out. You've been stimulating that muscle at some point for so long, even despite the slowness of that acetylcholine release, at some point you run out. Right? At some point, so many people have put coins in the vending machine and bought chocolate that at some point the stockroom is empty. And they just got to say, no more chocolate bars in the machine, right? And you just go hungry, right? And then you go off to lecture sad and hungry. But in any case, you got these three pools, right? So hopefully we can appreciate the relative role of each of these pools. What I want to do now is just get into a little bit of detail here in terms of how do we kind of ensure that these pools are kind of released in a certain way. So what they're trying to show us here in this figure is in green they've got the readily releasable and recycling pools kind of together as one. And then in blue they've got your reserve vesicles back here. Again, and I'll go back a slide here because I didn't really emphasize this perhaps as much as I should have. Note too that they didn't just draw these pools willy-nilly in space. The readily releasable ones are docked at the membrane. The recycling pool vesicles are just a little bit further back from the membrane, but the reserve vesicle pools are quite a bit further away. This drawing actually represents the spatial localization of these vesicles as well. And that's important here because what they're trying to show us is that if we stimulate this neuron with a low frequency stimulation, right? So maybe a low frequency stimulation or even just a weak stimulus that causes low frequency action potentials, we have these periods of depolarization that are triggering the opening of the voltage-gated calcium channels. But we also have these prolonged periods where the membrane is back to rest and where those voltage-gated calcium channels are going to close. So what we end up with in this low-frequency stimulation situation is we end up with just a little bit of calcium every once in a while coming in through the open voltage-gated calcium channels. And that keeps then any rise in calcium within this axon terminal pretty localized to the area adjacent to the presynaptic membrane. And again, I got to go back here maybe just to drive home a point that maybe I didn't drive home well enough. Oh, I'll go back further. How far back? Yeah. is to note here the location again of these voltage-gated calcium channels. Like they're located right down here, right, at the part of the presynaptic membrane that's facing the synaptic cleft. Like they're way down there. They're not way back up here where the reserve pool vesicles are. Those voltage-gated calcium channels are right down there, right adjacent to the synaptic cleft. They actually show that, I noticed as I was passing through, they show it right here too, right? There's our voltage-gated calcium channels. They're right down there where those readily releasable pool vesicles are, right? They're not way back up here where those reserve vesicles are. So when we have this low-frequency stimulation and we just periodically open up these voltage-gated calcium channels, we're going to get a little bit of calcium coming in, and it's going to mostly be localized to where the readily releasable and recycling pool vesicles are. So those are the only vesicles that are going to get signaled to be released. The reserve pool is not going to be signaled to be released. Whereas if we have a high-frequency stimulation, right, perhaps because we've subjected this neuron to a stronger stimulus, now because you're spending almost all of your time in a depolarized state, or you're certainly depolarized far more frequently, you're going to have those voltage-gated calcium channels opening far more often, perhaps even staying open most of the time here. And that lets a lot more calcium come into the axon terminal, and so more of it gets to that further distal region here where those reserve vesicles are, and then triggers them to start being released. So there's a strategic nature to the positioning of these different vesicle pools, right? You have your readily releasable ones right close to where the voltage-gated calcium channels are, your recycling pool just a little bit further away, and then your reserve pool quite a bit further away. And so that way, depending on the frequency of neuronal stimulation, which then determines how frequently the voltage-gated calcium channels are open, and therefore how much calcium can actually come into that axon terminal, the further away vesicles, those reserve pools, are only going to get triggered to be released if there's high-frequency stimulation, and therefore the voltage-gated calcium channels are open all the time, and therefore a lot of calcium is coming in. If you have low-frequency stimulation and the voltage-gated calcium channels are only open periodically and only a little calcium comes in, that calcium isn't going to get as far. It's only going to get to the releasable and recycling pool vesicles, and so only they will experience the signal that they should be released. Okay, let's talk now about the precise mechanism in terms of how exactly does calcium interact with these synaptic vesicles to cause their release. And we're going to start here by talking especially about the readily releasable pool, this pool that is docked to the presynaptic membrane. And what we're going to note here is our synaptic vesicle, right? And you can see the little blue neurotransmitters that are within it. And there are two proteins you can see here that are embedded into the vesicular membrane. One of these, which they've colored red, is synaptotagmin. And the other of these, which they've colored blue, is synaptobrevin. And it says here above that synaptobrevin is a snare protein, and it specifically is a V snare protein. The V here stands for vesicular, right? So this is the snare protein that is in the synaptic vesicle. There are two other snare proteins that are not in the vesicle, but instead are in the presynaptic membrane. One, colored in purple here, is SNAP25. The other in green is syntaxin. And these are referred to as T snares, and the T stands for target. These are target snares, and we call them target snares because this presynaptic membrane, where they are, that's the target for the synaptic vesicle. That's where that vesicle wants to wind up. Now, in terms of this idea of the vesicle being docked to the membrane, when we're thinking about this readily releasable pool, what causes this docking to occur is that all the snare proteins have an affinity for each other. And so the synaptobrevin that's present in the vesicular membrane is attracted to SNAP25 and syntaxin that are present in the presynaptic membrane, And so as they show in this diagram, all three of these snare proteins are kind of, attached together. And so because you have those two T snares that are in the presynaptic membrane, and you have the synaptobrevin of the vesicle attracted to them, the interaction between those three snare proteins is what's responsible for the docking of that vesicle of the readily releasable pool right there near that presynaptic membrane. Now, when the voltage-gated calcium channel gets triggered to come to open, and we've already established that will allow calcium to enter into the axon terminal, this is where synaptotagmin comes in, because synaptotagmin actually has two calcium binding domains in its structure. And so when calcium comes into the axon terminal, the calcium binds to those two calcium binding domains, and then at that point, synaptotagmin now has an affinity for two things it did not have an affinity for previously. One of those things is the T snares, those target snares that are in the presynaptic membrane. Synaptotagmin, once calcium binds to it, it suddenly wants to also join the snare party, so it wants to go to where those snares are, and those snares are in the presynaptic membrane. So that synaptotagmin moves towards the presynaptic membrane, and in doing so brings the vesicle right to the presynaptic membrane, which of course should facilitate the exocytosis of that vesicle and the release of those neurotransmitters. But the other interaction that forms now, once calcium binds to synaptotagmin, is calcium now also develops an affinity for phosphatidylserine. And if you're not sure what that is, that's a phospholipid. So here I have a typical neuronal membrane, and what you'll notice here is it's showing us a whole bunch of different phospholipids that are present in that membrane. In yellow is phosphatidylserine. And what you need to notice about phosphatidylserine is which half of the neuronal membrane do we find it in? It's in the inner leaflet only. It's on the cytosolic side of the plasma membrane only, right? It's not out there on the extracellular side. And so you have this phospholipid, this phosphatidylserine, that's present only in the inner cytosolic leaflet. And when calcium binds to synaptotagmin, synaptotagmin now wants to interact with phosphatidylserine. Well, because that phospholipid is in the membrane itself, this also is what brings the vesicle to that membrane to facilitate exocytosis. So it's this calcium-dependent attraction that synaptotagmin has for both T snares and phosphatidylserine. It's these two calcium-dependent attractions or interactions that facilitate this calcium-dependent exocytosis of these vesicles. Now, in terms of the attraction of phosphatidylserine, I'll point out here, because they highlight it in the figure, phosphatidylserine is negatively charged under physiological pH conditions. And of course, once calcium binds to the binding domains of synaptotagmin, because calcium is positively charged, it's that charge attraction between the bound calcium and the negative charge on phosphatidylserine. That's a big part of what attracts those two things together. I will also point out here, I added this note just for clarity, but there is also synaptotagmin present in the recycling and reserve pool vesicles, right? And so the calcium binding to the synaptotagmin of those vesicles is also critical for them to undergo this same exocytosis process. What I want to talk about next here is something you might be familiar with. Either you've experienced it personally or you've maybe just heard about it. But I want to talk a little bit about botulism, because it actually relates to the mechanism we were just talking about. Botulism is a foodborne illness, and you get it whenever you consume improperly canned foods. Anybody ever tried canning their own foods before? Okay, what did you can? What was it? A hummus? You did hummus? Okay, anybody else ever can their own food? What did you can? Yeah, okay, and it said try, so it didn't work out? They tasted okay. That's basically been my success with pickles, too. Okay, so there's a few little home canners around here, which is good. I wish I had more time to can things. But even if you buy canned food from the store, right, there's lots of canned food you can buy from the store, it's critical that that canning be done properly. Because if you improperly can the food, what will happen is a bacterium called Clostridium botulinum can actually thrive within that canned food if it's not canned properly, right? And so if you're a home canner, right, you're always like eating that pickle and thinking like, oh, I hope I did this right, right? Because if not, botulism is a real bitch. So what happens here? What is causing the botulism? How is it, being caused by this bacterium in this improperly canned food? Well, Clostridium botulinum produces a toxin. We call it the botulinum toxin. So it's this conglomeration of kind of different blue-colored things. LC stands for the light chain, and then the two H's stand for the heavy chain. So you kind of have these three protein chains that come together to make up the botulinum toxin. Now, the Clostridium botulinum makes this toxin, but in order for this toxin to have an effect on you, it actually has to get into your neurons. Now, it's kind of just like when we talked about rabies earlier in the course. Rabies also has to get into your neurons to make its way to the brain. And the way that rabies got in, we said, is it hijacked, right, that growth factor receptor. It basically, made itself look like a growth factor receptor, or made itself look like a growth factor, so it could bind to the growth factor receptor and trick the neuron into internalizing it. The same thing basically happens here. Right? So they've identified two proteins on the neuronal membrane that the botulinum toxin can bind to, and then once it binds, it triggers this internalization of the toxin. One, this SV2, is a protein we don't know a ton about, but the SV stands for synaptic vesicle. This is a protein that's present in the synaptic vesicle, and so when the synaptic vesicle undergoes exocytosis, that protein ends up getting into the neuronal presynaptic membrane, and then eventually, we want to internalize, that synaptic vesicle protein so we can use it again, and if it has a botulinum toxin attached to it, then we're going to end up taking that in with it. The other is this PSG here, which is like some sort of cell signaling protein or cell recognition protein. Not going to worry too much about it, but the idea, again, is that the botulinum toxin is binding to these proteins, right, that are normally present on the membrane surface, and then it gets internalized in that way, right, because the cell wants to internalize, especially that SV2, it wants to internalize that vesicular membrane that it sort of put in there during exocytosis. It wants to take that membrane back up at some point so it can use it to fill it with new neurotransmitters and dump them out again. So the botulinum toxin gets internalized by kind of hijacking this system. And then what happens here is once you bring this vesicle in, this endosome into the cell, we will often acidify endosomes as a way to try and get all the proteins in there to kind of come apart from each other so we can kind of recycle the proteins we want back to the membrane surface and kind of, destroy the ones we don't want. And that decrease in pH, that acidification, actually, the botulinum toxin uses that against us because that decrease in pH causes the heavy chain to basically open up a little pore in this endosomal membrane through which that light chain can then get out of the endosome and get into the cytoplasm of the neuron. And then this is where the trouble starts, because what they're showing us here at the end, this light chain from this botulinum toxin is actually an enzyme that chops up snare proteins. So if we go back for a second, snare proteins are critical to the neurotransmitter release process, as we identified. So if the botulinum toxin comes in here and chews up all the snare proteins inside of our neuron, then ultimately we can't release neurotransmitters. And so this is what the botulinum toxin does. And so one of the big problems is it causes paralysis. Because ultimately, in order to stimulate muscle contraction to allow for movement, we've got to be able to release acetylcholine in particular. right, to stimulate those skeletal muscles so they contract and we can move. But if you have botulinum and this toxin, chewing up all the snare proteins to prevent acetylcholine release from occurring, then ultimately you become paralyzed as a result. I'll end here with this final point, which is that clinically, we've actually exploited the botulinum toxin. And if you've ever taken or known someone who's taken Botox, and I'm sure none of us will admit to our Botox use. I mean, that's how I keep my youthful look here. But Botox is essentially the botulinum toxin being used in a clinical way. Because what we use botulinum toxin or Botox for is generally to paralyze facial muscles. You inject Botox into various regions around the face, as they indicate there in the picture. You're trying to inhibit the facial muscles from contracting, and that will help prevent wrinkling. Because wrinkling of the skin occurs when you have facial muscles contracting and therefore kind of constantly like moving the skin around. That's what causes wrinkling. That's why they tell you that you shouldn't smile if you don't want to wrinkle. Just keep your constant facial expression. But we inject Botox into your facial regions to actually get it to chew up the snare proteins in that region, and in doing so, inhibit facial muscle contraction from taking place, and then that facial muscle contraction being prevented will also prevent the wrinkles. As you're going to see in probably the practice quiz questions for sure, there are other ways that people are trying to use the botulinum toxin as well, because it doesn't just inhibit muscle contraction, it can be used to inhibit just neurotransmitter release more broadly. So anytime you want to shut down neurotransmitter release, you could do so using this toxin and the Botox. We'll stop there because the wolves are at the door, and we'll continue on Thursday with some more stuff about synapses.